Hello, and thanks for joining us today for the latest Technology Networks webinar, Expanding the Biologics Toolkit and Next Generation MALDI TopDown Sequencing. I'm Rebecca, a scientific content creator at Technology Networks, and I am here to moderate the event. I'm pleased to have Dr. Arndt Asperger from Bruker Daltonics joining us today as your presenter. Dr. Arndt Asperger is a senior application scientist at Bruker. He received his Ph.D. in analytical chemistry from the Institute of Analytical Chemistry, Leipzig University. He joined Bruker in Leipzig, Germany in 2002 before working at Bruker in Bremen, Germany in 2005. He worked on various positions, including product specialist and applications team leader on MALDI TOF/TOF. Currently, he is focused on MALDI-based pharma-related applications, including high-throughput screening. After the webinar, Dr. Asperger will be taking questions in a live Q&A session. You can submit questions for the Q&A session at any time during the presentation. Please enter any questions you have on the right-hand side of your screen and click send. For any questions we don't have time to get to, we will be sure you are contacted offline with an answer. Please remember, you can ask questions at any time, even while watching the webinar on demand. I'll now hand it over to Dr. Asperger to begin his presentation.
Thank you very much, Rebecca, for the nice introduction. Good morning, good afternoon, everybody. It's a pleasure to have the opportunity to present here in this Technology Networks webinar on the topic of characterization of biologics. And we have different methods. We have a bunch of methods available to characterize protein-based biologics. Some of them are using bottom-up strategies, others using top-down strategies. Today, I would like to focus on the top-down aspect of the characterization of biologics. And in particular, I would like to focus on a flavor of top-down sequencing that uses MALDI for ionization, a method that we used to call MALDI TopDown Sequencing, MALDI-TDS. For those of you who have not been using this technology, let me briefly introduce what actually MALDI TopDown Sequencing is all about and how it works very briefly. MALDI-TDS is a top-down method and looks at intact proteins. And to prepare such an intact protein for analysis, we prepare it in a certain MALDI matrix. We prefer to use super DHB as a MALDI matrix. We premix MALDI matrix and protein sample in a suitable ratio. That's what we always do when we prepare a sample for MALDI analysis. And then we spot this mixture on a MALDI sample plate and let it co-crystallize there, which gives us in the end a preparation, co-crystallized preparation spot, as you can see it on the photograph on this slide. Then we load the sample plate on the MALDI mass spectrometer and start the analysis by shooting a laser pulse, which triggers the ionization of the sample. And if we then look into the resulting spectrum, then we find that we do not just see signals for the molecular ion of the protein in the sample, but we also see additional signals which obviously originate from fragmentation of the protein in the MALDI ion source. And this is what we call MALDI in-source decay, MALDI ISD. And what kind of fragments do we actually see in such an experiment? We see both N-terminal and C-terminal fragments in the spectrum. The N-terminal fragments are of A and C type, whereas the C-terminal fragments are usually of Y and Z type. And these fragments are the product of different fragmentation mechanisms. A and Y ions are a product of thermally activated mechanism, whereas the C and Z type ions are the product of a radical-induced mechanism, which is pretty unique in ISD if you compare that, for example, to CID. Now, we can further extend this approach. That means we can actually select certain ISD fragments for further MS-MS analysis. That means we would isolate, for example, one of these ISD fragments and would then run an MS-MS experiment on this. And this approach is what we call T3 sequencing, which provides as additional explicit confirmation of the terminal sequence. Now, this method actually over recent years has proven itself to be really valuable for the characterization of protein biotherapeutics, and has been established for that purpose, which is reflected in a pretty large number of publications reporting on the contributions MALDI-TDS has provided to the analysis of protein tracks. I have just picked 3 examples on this slide. So it's kind of an established technique that turned out to be really valuable, really helpful in biopharma. Now, rather recently, we have actually launched a new instrument which has allowed us to further develop the MALDI TopDown sequencing approach. And we have now come to a new flavor of MALDI TopDown sequencing, which we call next generation MALDI TopDown sequencing. This is MALDI-TDS taking advantage of the technology that has been implemented in the new Bruker timsTOF instrument. I've just listed here the different key technology features of that new instrument. The timsTOF features a dual line source, which allows us to run both ESI and MALDI ionization. It's rather seamless switching between these 2 ionization modalities just via a software button. The instrument is equipped with a SmartBeam 3D laser system, which provides us unparalleled MALDI in-source decay performance. We can benefit from trapped ion mobility as an additional separation dimension for N and C terminal sequencing. And the instrument is featuring high resolution and high mass accuracy from TOF MS across a really wide M/Z range, and this yields us a really high confidence sequence readout. All of this we can now use to accelerate the characterization of protein biotherapeutics to get pretty instant data about the primary sequence, NNC terminal status, and near terminal modifications in biotherapeutical proteins. Now let's have a look on a couple of example data. Let's start with data obtained from a pretty well characterized model protein. For this purpose, we have selected the bovine carbonic anhydrase 2. This is a 29 kilodalton protein. And what we see here is the MALDI in-source decay spectrum of that protein. And there's one feature which is quite interesting in this spectrum, and this is that in such a MALDI-TDS spectrum, together with the C and N terminal sequence readout, we get actually delivered accurate intact mass information. As you can see here in this insets, we see doubly and triply charged molecular ion signals, which are nicely resolved into their isotopes and which are measured at extremely high accuracy, low to sub ppm accuracy. So that is a first very important information which we can get from such an experiment. And in a follow-up analysis of that spectrum, we can now read out the C and N terminal sequence in a very straightforward fashion from the MALDI ISD fragments which we detect in the spectrum. There is a couple of really unique things about MALDI in-source decay data, which I have listed here. So the sequence readout is particularly straightforward because the MALDI in-source decay fragments appear mainly singly charged. That means there is virtually no overlap between different charge series. That means we don't have to deal with a large number of different charge states. That simplifies the analysis of such data significantly. All the signals appear isotopically resolved, and we don't need to engage demanding algorithms for charge state deconvolution. We can just read the sequence from the left to the right in a linear fashion. So that is extremely convenient and makes data analysis simple and straightforward. In the inset, you have a closer look on the annotated spectrum. That means because of the high resolution the instrument provides, we can also achieve a very nice mass accuracy, because signal alignment and annotation is done very reliably. As you can see in this example, the RMS error across all the ISD fragments was approximately 1.7 ppm. And a further zoomed view on the higher M/Z region. So we see here in-source decay fragments from the N-terminus in the M/Z range beyond 10 kilodalton. And as you see, still all the fragments appear isotopically resolved. The peak finder does a very good job on annotation of monoisotopic signals. That means the annotated spectrum or the process spectrum can be directly read and can be matched against the sequence and gives us a very instant sequence readout. How about the sequence coverage we can achieve with such data? In this case, for bovine carbonic anhydrase 2, we get a sequence coverage which is close to 87%, which again is sustaining my previous statement that sequence readout is really straightforward. Compared to other fragmentation mechanisms like CID, for example, MALDI ISD proceeds as a statistical cleavage along the protein backbone. That means there is no really preferred cleavage sites, which also contributes to the high sequence coverage that we are able to achieve. I have shown you on the previous slide how impressive resolution and sequence readout is at the higher M/Z mass range. Now, of course, it's also interesting to have a look into the lower M/Z range, because that is actually important with regard to the question, how close can we get to the actual terminal? And that's why I have zoomed into a MALDI ISD spectrum here in the low M/Z range. I mean, in MALDI in general, you often have to deal with a pattern of background ions, matrix-related ions. That's why high resolution and high mass accuracy is particularly important in the low M/Z range to make sure that you are able to resolve your target signals from the background and also to assign and annotate the signals correctly. And this is obviously nicely possible with this data because of the resolution and mass accuracy it features. As you see here, we have multiple signals within a nominal mass, but we are able to nicely resolve these, and it's no problem to assign the respective fragments from either the N-terminus or the C-terminus of the protein, just because we can rely on the mass accuracy of the data. And that enables us to get really close to the terminus on both ends of the protein, N-terminus as well as the C-terminus. Now, of course, one of the key features of the timsTOF flags is its ability to engage ion mobility, trapped ion mobility, as an additional dimension of separation. And now we actually had a closer look on what can TIMS provide us in terms of benefits of for N and C terminal sequencing. That's quite an interesting question. And what you see on this slide on the right-hand side is actually a heat map plotting ion mobility versus M/Z. That means it's a MALDI in-source decay TIMS-MS experiment. And we see within the cloud of singly charged ions, we see a clear separation into two regions. They are referred to as R3 and R4 on this plot. And if we now have a closer look at these regions and what they actually represent, then it's a nice and very interesting finding that one of the regions is actually representing the in-source decay fragments from the N-terminus. That's why we see in the annotated spectrum the A and C type ions, whereas the other region represents the C-terminal fragments, Y and Z type ions. So that is, of course, of great help, in particular when you have to analyze data from proteins that are featuring unexpected sequences, or if you have to deal with even unknown protein sequences, because it means that you're able to dissect your multi-TDS data into terminus-specific portions, and it's then much easier to create sequence tag for either the N or the C terminus, and makes data analysis, simplifies data analysis significantly. There is another aspect where TIMS provides real benefit to top-down sequencing, and this is T3 sequencing, that means MS of selected in-source decay fragments. This is shown here on this slide for the N-terminal fragment C18, which you see in the spectrum, in the zoomed view spectrum here. This is the MALDI-TDS spectrum without TIMS separation. And as we see, the isotopic cluster of C18 is overlapping with another signal in the spectrum. That means if we would now isolate C18, we would co-isolate the overlapping isotopic signal cluster. But as soon as we engage ion mobility as an additional dimension of separation, we are able to separate these two ions, and then we can generate MS data that are kind of separated. It means there's an interference-free T3 spectrum of the N-terminal ISD fragment C18, which gives us explicit confirmation of the N-terminal sequence. So this was just to show you a little bit, or to give you a little bit of an impression how next generation MALDI top-down sequencing on the Bruker timsTOF flex works, what kind of data you can expect. And now I have put together two application examples, which I would like to present to you in the following slides. I would like to start with a first application case that deals with monoclonal antibodies. In this particular case, with the rapid sequence verification of subunits of adalimumab. The analysis of subunits is an established methodology for the analysis of antibodies. That means we would expose the antibody to a digestion step using an enzyme that's called fabricator. We would reduce the sample with TSAP, and then we would fractionate the sample by LC to get access to the separated subunits. We would collect those fractions in Eppendorf tubes or in a microtiter plate, and would spot the fractions that contain the pre-separated antibody subunits on a MALDI sample plate, which we would then use to analyze it by MALDI top-down sequencing on the timsTOF flex. And here we see the results in a nutshell that we obtained from such an analysis. On the left-hand side, we see a table which shows us the key performance parameters that we achieved for the adalimumab subunits. We got intact mass information at an accuracy level of below 1 ppm based on the doubly charged ions. And we got a sequence verification percentage for all three subunits in the range between 70 and more than 80%, depending on the individual subunit. So that is quite nice, and it's a pretty instant information we get. To all of this information that you see on this slide is actually provided in 3 spectra, right? It's just a minimum amount of data providing a pretty large amount of information. So that's certainly also one very unique feature of MALDI top-down sequencing as an approach. Just to give you a little bit of more insight into the raw data, this is from the FC subunit, taking into account the major glycoform G0F. Adalimumab has a rather low heterogeneity glycosylation and glycosylation pattern. That's why we decided not to perform a deglycosylation step, but we could use the sample as is. And the in-source decay spectrum you see on the right-hand side has been matched again the most abundant glycoform G0F. Again, as I said showed on the previous slide, we get this nice confirmation of the intact mass of the subunit by means of this high resolution data from the doubly charged molecular ion, and then we see the sequence readout from both the C and N terminus, nicely resolved spectra, highly accurate, and yeah, it's a very fast technique to match, to verify the sequence of your target protein. On this slide, I have picked a few examples from the LC spectrum to show you the importance, actually, or to illustrate once again the importance of resolution and mass accuracy for correct sequence annotation. I mean, the MALDI ISD spectra can be pretty complex, as you have seen also on the previous slide. And that's why it's very important that we can resolve all these signals that are showing up in such a spectrum. Sometimes peaks or fragment ions originating from the opposite termini are very similar in mass, and it's really important to be able to resolve them. As you see, we can resolve them very, very nicely, as you see on these examples. But of course, sometimes you may run into cases where certain fragments are really overlapping and where you don't have a chance anymore to resolve this by mass spectrometry alone. And then, of course, again, it's great to have ion mobility as an additional separation dimension, because then we can check by ion mobility if this signal is really a composite signal accommodating both. And as you see, we get proof for the presence of both of these signals from either the C terminus or the N terminus as a Y and A ion, just a tiny little difference in mass, but a clear difference in mobility allows us to separate these two. So as a second application example, I have picked something that is rather closely related to the situation in which we are currently. I mean, we are all feeling still the pandemic situation every day. And the following example data are actually from a project that was about the characterization of antigens, actually about the characterization of recombinant SARS-CoV-2 as glycoprotein RBD expression products. So for the development of vaccines, it's really important to have access to well-characterized antigens. And this is what we have been looking at in this example. The samples actually came from actually 2 RBDs expressed in either Chinese hamster ovary cells or in human embryonic kidney cells. They have been produced by in vivo biotech services. We exposed these samples to an enzymatic deglycosylation step in the beginning, where we actually released by PNGase F the N-glycans. Then we removed the sialic acids with a CL-AXO from Genovis, an enzyme from Genovis. And optionally, we also removed the core-1 O-glycans with O-glysol. In the final step, we applied DTT to reduce the sample. And then we applied next generation MALDI TopDown sequencing on timsTOF to analyze the O and N glycosylation in these antigens, actually, to get a better clue of the type of glycosylation and also glycosylation sites. And here's what we've got in these experiments. Let's first have a look on the type of O glycosylation present on either of these 2 RBDs. In the CHO RBD, there we can tell from the intact molecular ion signals we got, we could actually confirm the expected amino acid sequence. And at the same time, we could safely say that there's obviously a core-1 O glycosylation attached. Considering this core-1 O glycosylation, a single core-1 O glycosylation, we could confirm the expected mass with an accuracy of 1.4 ppm. So that's great. That means it's meeting all the expectations of the antigen product. For the HEK RBD, the situation was a bit more complicated. Actually, what we could confirm or verify was that this was actually a modified amino acid. That means there was an N-terminal pyroglue extension that came from an unexpected cleavage within the signaling peptide. This has been found actually earlier, and we just verified it with our data. This has been published in Analytical Chemistry rather recently. And with regard to the type of O glycosylation, we could also detect a core-1 O glycosylation, a single O glycosylation. But in parallel, we saw a second signal, and that refers obviously to core-2 O glycosylation, which is present in parallel. That means in HEK RBD, there we have both core-1 and core-2 O glycosylation present. Now we performed another enzymatic deglycosylation experiment using O-glysol just to confirm once again that the core2 O-glycosylation was actually there in the HEK product. The O-glycol enzyme removes core1 O-glycosylations, and core2 O-glycosylations are resistant to this enzyme, and this gave us final proof that in fact we have both core1 and core2 in the HEK RBD. As a next question, we then wanted to know where are actually the active O-glycosylation sites. And for this, we performed an in-source decay experiment. And what you see here on this slide is actually a zoomed view on the MALDI ISD-MS spectrum from the CHO RBD, in which we can clearly see that we get a match of the sequence if we consider T5 as an active site of core1 O-glycosylation. That means the in-source decay spectrum gives us clear proof for T5 as the active site of core1 O-glycosylation. We can further prove this by performing T3 sequencing. That means we selected the N-terminal fragment C6 for further fragmentation by MS-MS, CID MS-MS, and in the resulting T3 spectrum, we find the diagnostic fragments which we would expect for the fragmentation of a hexose and hexamer unit. That means this gave us another final proof for the fact that T5 is the active site of core1 O-glycosylation in this expression product. We performed the same type of analysis also for the human embryonic kidney cell RBD. And there we find that T6, so this basically the same glycosylation site is active site of core1 O-glycosylation, again confirmed by T3 sequencing, and we also performed the matching of the data against the sequence modified with the core2 O-glycosylation, and again we could verify that threonine at 6 position was the active site of core2 O-glycosylation. The serine at position 8 is obviously not glycosylated. Finally, we also had a look at the N-glycosylation aspect, and for this we performed a MALDI ISD TIMS-MS experiment. And what I would like to show you here is that TIMS offers some nice separation features which can help us to answer multiple questions within one and the same analysis. What you see in this upper plots, these are the heat maps, TIMS-MS heat maps for either of the two expression products. And there in both of them, we see actually two regions of ion populations. R1 is the cloud of in-source decay fragments of the protein backbone, and R2 is actually not related to the ISD fragments or to the protein backbone, but it is actually related to the N-glycans that are in the sample which have been released by PNJSF. That means from the in-source decay spectrum of the protein backbone, we can nicely confirm the N-glycosylation sites just by matching the ISD fragments against the sequence and considering the modified asparagine residues that have been induced by the PNJSF treatment. And in the bottom part, you see the extracted spectrum comprising the different N-glycans, so giving us a complete N-glycan compositional profile from the very same data set. That means one experiment, MALDI ISD TIMS-MS experiment, gives us both confirmation of glycosylation and glycosylation sites, as well as the composition profile of the N-glycans themselves. One interesting finding here was that in the HEK RBD, we find a distinct difference in the N-glycosylation profile compared to the CHO RBD. The expression product from the human cell lines contains a significant amount of multiply fucosylated species, which we then also confirmed by MS-MS. That means these additional MS-MS spectra give us proof of the structure which contains multiple fructose residues. Okay, great. Let me briefly summarize what we have learned from the data that I showed before. I hope I could convince you that next generation MALDI top-down sequencing is really a highly efficient method accelerating the characterization of protein biotherapeutics. Next-gen MALDI-TDS provides instant high confidence information delivers this information regarding primary sequence, C and N terminal status, and near terminal modifications, for example, N and O glycosylation, as we have seen from the previous example. N and C terminal sequencing benefit from trapped ion mobility spectrometry in combination with high resolution, highly accurate TOF-MS. TIMS enables dissection of terminus-specific MALDI-TDS spectra, simplifying data interpretation, and it also enhances T3 sequencing for explicit verification of terminal sequences. Finally, TIMS also enables the confirmation of N-glycosylation sites and N-glycan profiling in a single MALDI-TDS analysis. In MALDI-TDS, the sequence readout is extremely straightforward due to the fact that we are dealing with singly charged MALDI ISD fragments mainly. That means we don't need to engage charge deconvolution. And we get provided a wealth of information in a minimum amount of data. And finally, I would like to stress that actually MALDI-TDS means a really nice add-on to the unique top-down sequencing capabilities in the timsTOF Biologics Toolkit, which can engage both ESI and MALDI workflows. If you would like to read more about the topic we have been talking here in this webinar, then please, you are invited to go to the Bruker website and to download from there the literature I've cited here. We have a couple of application notes there on this topic, and also from the Bruker ASMS 2021 poster hall. Please have a look on poster TP271, which is providing more details on the SARS-CoV-2 story. I have been mentioning that actually MALDI-TDS, it's one additional tool in the toolbox the Bruker timsTOF is offering for the characterization of biologics. And that actually coming back to that brings me to this slide. I would like to show you just a few examples for other biologics beyond proteins. That means the instrument, of course, can be used very nicely to analyze other biologics, for example, oligonucleotides and RNA. So what you see here is actually data from oligonucleotides of different sizes, so from the 10 mer as well as the 25 mer. As you see, we get very nice high resolution and high accuracy data from these oligonucleotides when we run MALDI-based QC analyses at really high speed and throughput. So whenever throughput matters, MALDI, of course, is a very nice option. And another detail which is really interesting is that we are indeed able, by means of timsMS, to also discriminate isomeric oligonucleotides. That is shown here for four oligonucleotide, which comes here as 4 different isomers. As you see in the ion mobility trace, they are all distinct, showing distinct profiles, are all different, and it's no problem to distinguish these different isomers based on the mobility data. So that is, of course, a really nice, exciting, and new application of ion mobility for oligonucleotides. Despite of MALDI, of course, there are certain workflows where you would prefer to use ESI, LCMS based workflows. That's why I've picked up here also this. That means we can also analyze oligos by LCMS, even larger species that are in the size range of up to 30-35 kilodalton, as you see on this slide. And the LCMS-based analysis would benefit from a couple of new features that are present there on the timsTOF, like the vacuum insulated probe heated electrospray interface, the ultra high resolution the instrument is offering, passive MSMS, and finally also a software aspect comes into play here. That means there is a new software available called OligoQuest, which is very, very supportive in the annotation of MS and MS data from oligos and RNA samples. And my very last slide is just summarizing once again what I said before, the integration aspect. The timsTOF Flex is not a one-trick pony, but is extremely versatile and offers a broad toolkit for the characterization of biologics based on either ESI or MALDI workflows. CCS aware, that means engaging ion mobility as an additional dimension of separation. And yeah, I think it's setting a new standard for the characterization of biologics. That was my last slide, actually. Thank you very much for your attention. And if you have any questions, please let me know and I'll be happy to answer them. Thank you very much.
Thank you very much, Dr. Asperger. I'm sure you'll agree that there were some excellent points raised during your presentation. We will now begin the Q&A session. To submit a question to our speakers, you should enter your questions into the box on the right-hand side of the screen and click send. We will answer as many questions as possible during the time available. We've had several questions submitted, so I'll move straight on to these. So first question, can the MALDI top-down sequencing be utilized to identify antibodies related to infectious diseases?
Well, that depends a little bit on the context of the analysis. In principle, of course, it can be done, but the key question here is probably how and or if you can get access to a sufficiently large amount of such an antibody. You would need to isolate it somehow because in MALDI top-down sequencing, we fragment a protein in the ion source directly. That means we do not isolate a particular species, but it's a fragment all technique. And therefore, we need to isolate a protein first before we can then analyze it by MALDI top-down sequencing. So answer is yes, in principle, you could think about that. But first, you need to find a way to isolate the protein, the antibody, in a sufficiently large quantity. How much material do we need for an analysis? I would say an amount of 10 to 20 picomole is required per analysis to obtain decent quality data in the end.
Thank you very much. So next question, what's the upper molecular weight limit for proteins to be analyzed with this method? Is there any?
In principle, there's no limit for molecular weight of proteins to be analyzed with MALDI top-down sequencing. That means we can obtain terminal sequences from MALDI-TDS data for very large proteins. What, of course, is different is the overall sequence coverage you can achieve. The larger the protein, the more the overall sequence coverage, of course, will drop. This is simply because of the fact that what we observe in the spectra is terminal fragments, and the more you get into the center of the sequence of the protein, the lower the chance that you can still detect those fragments. It's just a matter of the accessible M/Z range in such an analysis. But in principle, there's no limit in molecular weight. It's all about the sequence coverage in the end.
Fantastic. So what's typically the amount of protein you would need for analysis to obtain decent quality MALDI ISD data on timsTOF Flex?
Okay, that's kind of redundant to the question we had before. I would say from experience, I would say typically we use a lower double-digit picomolar amount per analysis, so 10 to 20 picomole per analysis is what we typically use.
Fantastic. So is there any software available supporting MALDI ISD data interpretation?
Yes, certainly there's software available. Otherwise, we would certainly not be able to extract the information that such a spectrum contains to full extent. So we actually offer two software tools that can analyze MALDI-TDS data. One software is more focusing on the routine aspect of data analysis, let's say a QC type of analysis, so straightforward matching of MALDI ISD data against target sequences, eventually differently modified versions of a target sequence to figure out if either the one or the other or both modified versions contribute to the spectrum match. So this is the software is called Biopharma Compass that can do that rather routinely, also for larger batches of TDS data sets. Alternatively, if you need to dive deeper into your data, for example, because you run into cases where an unexpected modification or an amino acid exchange is making the analysis more troublesome, then you would eventually use the Bruker BioTools software, because there you can engage additional tools like, for example, top-down mascot database searching, or also a kind of sort of complete de novo type of analysis. So that's all covered in the Bruker BioTools software.
Brilliant. Thank you very much. So can MALDI-TDS deal with protein mixtures? If I understand correctly, it's a fragment or method. So which level of complexity can it deal with without the need for engaging LC pre-separation?
Yeah, that's an interesting question. I think part of the answer I gave already in one of the earlier questions, but anyway, it has a couple of aspects. I think in the presentation we have seen two cases where complexity mattered. Actually, one was the subunits of the antibodies. There we indeed applied a separation step, a LC separation step, simply because otherwise, if we would have all three subunits in a mixture, the resulting MALDI-TDS data would simply be too complex, and we would lose information just because of the level of complexity we would no longer be able to deal with. On the other hand, we have seen the SARS-RBD case, where we had actually differently glycosylated versions of the same protein primer sequence in one and the same sample, and there the complexity was at a level which we indeed were able to deal with, because it was just one single amino acid that was differently modified. And this is something you can certainly deal with, that you have multiple C or N-terminal fragment series, which are just different in one amino acid or one modification. That's something you can deal with. If you really have mixtures of proteins that would all give you individual C and N-terminal fragment ion series, then probably you come to the point where you need to think about separation prior to MALDI-TDS analysis.
Fantastic, thank you. So you mentioned sthb as a preferred MALDI matrix for top-down sequencing by MALDI ISD. What's actually the difference between sthb and regular DHB? Would DHB work either?
Probably DHB would give you also results. Our experience is that super DHB works even better than DHB. In general, super DHB these days is our clear preference for MALDI-TDS analysis as a MALDI matrix for TDS analysis. The difference between DHB and super DHB is that super DHB is actually a mixture of two different molecules. It contains as a major component just normal DHB, but in addition, it contains a certain portion of the monomethylated form of DHB. That means this would be 2-hydroxy-5-methoxy hydroxybenzoic acid. So it's a slightly modified version or chemically modified version of DHB. And in our hands, it is the best working matrix for MALDI top-down sequencing analyses. Both DHB and super DHB can be purchased from commercial vendors. They are both available as products.
Thank you very much, Dr. Asperger. So the last question, can you accumulate the data in order to increase SN, especially in the higher molecular weight region, and so increase the length of the sequence?
Of course, we are free to accumulate data from as many laser shots as we want. I mean, the spectra which we have seen on the slides are, I would say on average, they have been accumulated from thousands of laser shots, which is not a big deal with regard to time you need for acquisition, because we are using a laser system that features a repetition rate of 10 kilohertz. So that means in the end, this is a matter of seconds. What we do, in fact, is that we sometimes acquire spectra specifically from certain positions on the sample crystallization spot, when it turns out that you have spots that work particularly well and other raster positions that don't. So we can be very selective, and we are free to accumulate the spectrum from a large number of spots to get better signal. Indeed, that's what we do. Yeah, that's what how I can answer this question. With regard to the higher M/Z mass range, I have to say that the fact that the resolution is extremely high also for these large M/Z ions, that helps us a lot, even for signals that do not feature a particularly high signal-to-noise ratio. The resolution can help there a lot, because it makes it much easier for the peak finder, if you have nicely baseline resolved signals, to de-isotope those signals and to give you the correct monoisotopic mass for such a high mass fragment. And as you have seen, this gives us very nice and long stretches of sequence readout. And this was all based on automatic peak finder using a de-isotoping peak finder like the SNAP peak finder in the Bruker software. So that's pretty amazing how that works if your data is sufficiently well resolved.
Brilliant. So thank you very much. That's all we have time for today. Just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. You can continue to ask questions even when watching the webinar on demand. A certificate of attendance is available to download from the Handouts tab on the right-hand side of the webinar platform. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar, please feel free to share the link. Thanks again for your time.